also continue the theme of oviposition traits in sawflies. And I'm going to talk about selection on oviposition traits and how that generates reproductive isolation between two pine sawflies. So one of the fundamental goals of evolutionary biology is to understand how new species arise. And one mechanism that is important in generating biodiversity is ecological speciation. In ecological speciation, you start out with an ancestral population. This population then moves to a new environment. And in this new environment, natural selection favors different traits than in the ancestral environment. And this divergent natural selection causes reproductive barriers to arise. However, in many cases of ecological speciation, we don't know the nature of the traits under divergent selection, nor the mechanism connecting divergent selection to reproductive isolation. One of the unique barriers in ecological speciation is extrinsic post-zygotic isolation, which is where you have reduced hybrid fitness in the parental environments due to intermediacy of traits. And one of the classical examples of that is found in stickleback fish, with the limnetic and benthic forms having different mouth morphologies that are respective to their feeding strategies. And the hybrids have an intermediate morphology that causes them to be less successful in feeding. However, this form of reproductive isolation is understudied compared to other forms, partially because it can be hard to distinguish from intrinsic post-zygotic isolation or genetic incompatibilities. So to study this, I'm also using Neodypyron sawflies. Neodypyron sawflies are pine specialists, and they have adaptations throughout their life cycle to deal with pine resin, which is a quite nasty substance. First, the females have saw-like ovipositors that they use to cut a pocket into the needle, and then they insert their eggs into that pocket. And throughout the genus, all of the species have a wide variety of strategies to deal with oviposition. Additionally, as larvae, they have feeding adaptations. You can see here in this photo where a small instar larvae actually eats around the edge of the needle so that they don't eat into the pine resin canal. Whereas later instar larvae actually sequester the resin so they can use it later as a defensive mechanism by regurgitating it. And specifically, I was using two sister species that vary in their host use, Neodypyron pinetum and Neodypyron lecontii. Pinetum is a white pine specialist, whereas, as you've just heard, lecontii is more of a generalist that uses a wide range of hosts. However, it does not use white pine. And we think that adaptation to white pine is what actually drove these two species apart. And to understand how that could happen, we have to actually look at how the hosts themselves differ. And one of the ways in which they differ is in their needle architecture. And this includes a large amount of variation in needle width. So to just confirm this, I went out to the field, collected a bunch of measurements of pine needle widths for a bunch of species, and throughout this talk, white will refer to pinetum and pinetum's traits, whereas yellow will refer to lecontii and lecontii's traits. And we found that white pine is a much thinner needle than lecontii's host, and that is important because of how they oviposit into that needle. If they cut too deeply into the needle will dry out, causing the eggs to dry out and die. On the other hand, if they don't cut deep enough, their eggs can get inundated with resin and they'll drown. Additionally, there is evidence of hybridization between pinetum and lecontii in nature. First, we found that there's mitochondrial integration, and second, we've collected hybrids in the field. And since these two species remain distinct despite hybridization, we suspect that there is some form of post-zygotic isolation keeping these two species apart. Which leads us to our hypothesis that variation in needle width generates divergent selection on oviposition traits and causes reduced hybrid fitness. So to test this hypothesis, we have a series of predictions. First, if oviposition traits are under divergent selection, females will have morphological and behavioral oviposition traits that match the needle architecture of their host. And to test this prediction, we placed a mated female into a choice cage with 2 white pine and 2 jack pine seedlings. The female was then allowed to oviposit. And we see that, as expected, pinetum prefers white pine and lecontii does not, and this preference is a very strong preference. The next behavior that we were interested in was oviposition pattern. Pinedum lays about 1 to 3 eggs per needle, whereas Lecontii lays many more eggs per needle. Additionally, Pinedum's eggs are more widely spaced than Lecontii's. And the final behavior we were interested in was the formation of testlets. So the testlet is simply just a small cut in the side of the needle before the eggs are laid, and this testlet allows resin to drain from the needle, and you can actually see in this lower testlet here a droplet of resin coming out of that needle. And we find that Pinedum never actually cuts a testlet, whereas Lecontii is pretty consistent in cutting testlets. We were also interested in ovipositor morphology and egg morphology in addition to behavior. And we find that Pinedum has both shorter and narrow ovipositors. We then used geometric morphometrics to look at the shape differences between Pinedum and Lecontii's ovipositors. And this is, right here, is the plot from the principal components analysis, with the work grids down here showing the change in shape along PC axis 1. And that change in shape really refers to the curvature of the ovipositor. And you can see that the Lecontii ovipositors all cluster over here, and Pinedum's all cluster over here, with Pinedum having a much straighter ovipositor than Lecontii. Additionally, Pinedum has shorter eggs than Lecontii. So our first prediction is met. Lecontii seems to have traits that are well suited for ovipositing into a thick needle, whereas Pinedum seems to have traits that are well suited for thin-needled hosts. Next, if there's extrinsic postzygotic isolation, we expect that hybrids will have reduced fitness, and this reduction in fitness will be host-dependent. As I mentioned earlier, it can be difficult to distinguish from intrinsic and extrinsic postzygotic isolation, but one way in which you can do that is through reciprocal transplants of the back crosses. And if there is extrinsic postzygotic isolation, you expect there to be an interaction between host type and cross, with the back cross having the highest fitness in the parental environment to which it's most genetically similar. And you can see that here, for example, with the Lecontii back cross shown in blue having a higher fitness on Jack than on white pine, and the opposite pattern with Pinedum back crosses. So here is our cross design. We took females of both species and mated them with a conspecific male. We then took the F1 females and mated into the haploid males from the same cross. It may be important to know for you that don't know hemipterans, they have haplodiploid sex determination. And these crosses then generated back cross sets, one in which you had a mixture of alleles on a Lecontii background, and another in which you had a mixture of alleles on a Pinedum background. And the first trait we were interested in was simply host preference, and we found that hybrids actually prefer white pine, and at least in this experiment, Lecontii never oviposited on white pine. We then looked at the fitness of these crosses, and since we were interested in the effect of oviposition traits on fitness, we used oviposition success as a metric for fitness. And we defined oviposition success just just simply if a female had eggs that hatched. And as you can see, the hybrids do have reduced fitness, suggesting that there is postzygotic isolation. Next, to see if this reduction in fitness is host-dependent, we separated out the cross types by the host the female oviposited on. And we didn't find the interaction we were expecting. However, the reduction in fitness is still host-dependent. On jack pine, all of the cross types have equal fitness, whereas on white pine, the hybrids and Lecontii back crosses have reduced fitness compared to Pinedum. So overall, this does suggest that there is extrinsic postzygotic isolation. So our second prediction was met, and our third prediction was partially met. Finally, if oviposition traits are responsible for generating reproductive isolation, we expect back crosses on white pine will have higher fitness if they have Pinedum-like traits. And to test that, that, we looked specifically at Leconte back crosses on white pine. And the first trait we looked at was oviposition pattern. And we used a qualitative categories for this, with a pineatum pattern being 3 or less eggs per needle that were widely spaced, whereas any other pattern was considered non-pineatum-like. And we did find that individual females that had a pineatum pattern had greater success in hatching than those that did not. And the next trait we looked at was ovipositor morphology, and we found that females that had eggs that hatched had shorter ovipositors. So overall, our fourth prediction was met. So in conclusion, there is selection on oviposition traits, and this includes both behavior and morphological traits. And the selection results in extrinsic postzygotic isolation. And our future directions include Q-tail mapping of oviposition phenotypes to find the loci underlying these traits, as well as genome-wide scans for regions of differentiation and signatures of selection. Additionally, we want to carry out comparative analyses of ovipositors across the genomes to see more broadly how ovipositor morphology relates to host use. And I would like to acknowledge all of the members of my lab that helped me on this project and the funding sources, and if anyone has any questions.